In a darkened clinical room, a patient is prepped for a procedure. A doctor carefully aligns a specialized fiber optic cable and flicks a switch, sending a pure red beam of light across the space. From early sun-based treatments to high-contrast imaging, light has been a recurring driver of medical development. But today, it serves a highly specific, offensive function. In photodynamic therapy, this beam operates as a scalpel of photons. It is a tool for microscopic precision. A single beam awakens a dormant agent already resting inside the patient. This turns chemistry into choreography, unleashing a cascade of reactions that targets disease at its core. This process creates a synthesis of science and light, where structural healing begins with a single spark. To see why this intervention is necessary, look at the primary obstacle in treating localized diseases like cancer: the problem of collateral damage. This cellular map illustrates the challenge. Traditional systemic treatments and wide surgical excisions often act as blunt instruments. They destroy the diseased tissue, but their treatment zones inevitably overlap with the surrounding healthy cells. Because these therapies cannot fully distinguish between normal and abnormal tissue, they take a heavy physical toll on the body. The patient is often forced to recover from the cure itself. This created a requirement for a treatment method that could differentiate friend from foe at a microscopic level, a way to target cells individually. To achieve this, researchers moved the focus down to the cellular level, using light as the trigger for a localized reaction. This approach prioritizes localized destruction, removing the threat without disturbing the rest of the body. Deep within the microscopic landscape of human tissue, cells are constantly absorbing the various compounds that move through the surrounding fluids. The first functional step of photodynamic therapy is to introduce a specialized photosensitizing drug, a dormant agent, into the patient's bloodstream. This chemical circulates through the body, remaining inert and harmless as long as it is kept in the dark. Over a period of hours, the agent naturally filters out of normal tissue. However, because diseased cells metabolize differently, the drug becomes heavily trapped inside the abnormal cells, building into a high concentration. By itself, this compound does not attack the cell. It sits as a positioned molecular trap, waiting for an external trigger to activate it. That trigger is a laser, tuned to a specific wavelength and aimed directly at the area of the disease. In this tissue cross-section, light penetrates exactly to the depth of the agent-filled cells. Photons strike the accumulated molecules, splitting ambient oxygen into highly volatile reactive oxygen species. These destroy the abnormal cell from the inside out, leaving adjacent healthy cells entirely intact. This destruction is strictly bound to a triad. The effect only occurs where the targeted cell, the specific drug, and the calibrated light all intersect. Moving back out to the macro scale, the clinical result of this cellular process is immediately visible. The diseased tissue is removed, but the patient completely avoids the broad systemic sickness typically associated with circulating active toxins. Instead of a recovery period defined by healing from the treatment itself, the patient experiences a more localized, precise recovery. This demonstrates how light, when tuned to a specific wavelength, can serve as a precision trigger for biological change. It shifts the strategy away from flooding the system toward engineering targeted drugs that activate only when needed. Ultimately, photodynamic therapy demonstrates the true potential of targeted medicine, where profound healing. Modern medicine faces a biological stalemate. As our treatments escalate in toxicity, diseases adapt. Tumors learn to hide by camouflaging their surface proteins. Bacterial colonies secrete impenetrable physical walls. And in the lab, rogue stem cells mutate into dangerous growths. The standard response is to attack with even harsher chemicals. But an alternative strategy bypasses the chemical arms race entirely. It relies on a precise, targeted application of light to dismantle these biological shields from the inside out. This process is called photodynamic therapy, or PDT. It relies on a three-part mechanism. First, a light-sensitive drug, a photosensitizer, is introduced. When hit by specific wavelengths of light, this dormant drug activates, reacting with local oxygen to generate a burst of highly reactive molecules called reactive oxygen species. In 2026, researchers have learned to harness this oxidative stress as a molecular switch. These reactive pulses function as secondary messengers, traveling deep into the cell's signaling pathways. Instead of causing random damage, they trigger a specific genetic cascade that prompts the cell to dismantle its own defensive walls. We are shifting away from blunt force trauma and moving toward targeted biological hacking. By decoding these light-activated signals, we can systematically defeat 3 of medicine's most complex diseases. In oncology, a tumor's greatest asset is its ability to hide. When stressed, cancer cells accelerate an internal recycling process called late-stage autophagy. This process breaks down MHC1 molecules, the critical chemical flags that sit on a cell's surface and tell the immune system what to attack. Without these flags, the tumor becomes completely invisible to the body. To counter this, modern photodynamic protocols introduce a specific chemical roadblock. Clinicians pair the light therapy with chloroquine, an existing drug that effectively paralyzes this late-stage recycling process. Halting the cell's internal recycling forces the tumor to keep its MHC1 flags prominently displayed on its surface. The cancer can no longer hide. This intervention releases the brakes on the immune system, ensuring the tumor remains completely visible during the photodynamic attack. Clinicians amplify this visibility through a process called immunopriming. Pre-treatment with vitamin D prior to the light therapy aggressively alters the T-cell population. It reduces the number of exhausted, suppressed immune cells and actively recruits fresh cytotoxic T-cells to the treatment site. This highly visible, localized cell death releases a specific blend of chemokines into the bloodstream. These chemokines create a molecular trail, guiding immune cells out of systemic circulation and pulling them directly toward the cancer. Because the immune system is now trained on the tumor-specific markers, it begins hunting down secondary cancer sites in entirely untreated organs. A highly targeted beam of light, combined with careful chemical priming, coordinates a permanent, body-wide immune cascade. Bacteria survive our medical interventions through sheer physical fortification. Multi-drug-resistant strains construct an EPS matrix, a dense structural web that encases the colony. This biofilm physically blocks drugs from reaching the bacteria, multiplying their antibiotic tolerance by up to 1,000 times. Antimicrobial photodynamic therapy acts as a physical wrecking ball against this defense. The reactive oxygen species generated by the light physically sever the bonds of the biofilm. The matrix collapses, exposing the underlying bacteria to severe, immediate oxidative stress. But stripping the physical armor is only the first step. The treatment also deploys a precise biochemical attack. Light-activated metal complexes, known as Ru1, are introduced specifically to hunt down and neutralize NDM1, the specific enzyme that gives these bacteria their chemical resistance to drugs. The clinical data confirms the efficacy of this two-pronged assault. In a landmark 2025 study from the Crick Institute, neutralizing that single resistance enzyme with light-activated complexes caused a massive shift in vulnerability. The common antibiotic meropenem became 53 times more effective at destroying resistant strains of E. coli. By shattering the physical biofilm and biochemically disarming the resistance enzymes, light therapy strips the superbug of its defenses, allowing our failing antibiotics to become lethal again. Beyond oncology and infectious disease, light acts as a strict regulator in regenerative medicine. When scientists grow new tissues from pluripotent stem cells, they face a severe risk. If any cells remain undifferentiated, their blank slate status allows them to mutate and form dangerous tumors, known as teratomas, inside the patient. To prevent this, researchers use the CDY1 probe. This highly specialized marker is engineered to sweep through a developing tissue sample, seeking out and attaching itself exclusively to those lingering, undifferentiated ORG cells. This initiates a process called extrinsic photobiomodulation. When the tissue is exposed to targeted light, the CDY1 probe activates. It ignites a burst of targeted reactive oxygen species directly inside the mitochondria of the rogue cells. The undifferentiated cells are destroyed on the spot, while the surrounding healthy specialized tissue remains completely untouched. Light functions as an intelligent quality control mechanism. It systematically purges the dangerous outliers, ensuring that the lab-grown organs we eventually implant are genetically validated, structurally functional, and completely safe. We have moved past the era of treating disease with indiscriminate chemical saturation. By mastering the precise application of controlled oxidative stress, we can actively program cellular fidelity and dictate long-term immune memory. This molecular understanding is now shifting the physical architecture of our hospitals. A growing discipline known as chronoimmunotherapy is reviving the historical concept of open-air medicine, leading modern clinics to build integrated outdoor treatment environments known as healing landscapes. These therapeutic gardens utilize daylight photodynamic therapy. Instead of relying entirely on artificial lasers, the open-air design naturally captures the 11,000+ lux of sunlight required to activate photosensitizing drugs smoothly and painlessly. This environmental shift is driven by strict biological necessity. Aligning the administration of light with the patient's endogenous circadian rhythms synchronizes their core clock genes. This synchronization actively accelerates dendritic cell migration, optimizes the necessary autophagy cycles, and significantly improves the body's overall healing response. Photodynamic protocols have transitioned from simple localized procedures into tools for complete biological synchronization. This approach integrates molecular science with natural rhythms, proving that medical healing can begin with a single spark of light.